Our next speaker is Dr. Vamsi Veeramachaneni, and he is from Strand Life Sciences. He speaks to us on identifying large variations in the human genome by next generation sequencing. Dr. Vamsi.
Morning, everyone. Thank you all for this invitation, Chirag. Pleasure to be here. As some of you may know, the company Strand is probably around 24 years now, and it got incubated out of ISC. So it's a pleasure to be here giving this talk. Let me start. Just a show of hands, how many of you are from the computer science background here? Data science and computer science? Small fraction. So, and from the biological sciences? Okay, yeah, thanks. Gives a context. I'll start talking about copy number detection in clinical samples. I think somebody asked Bratati the question before about how do you actually go about identifying these variants and what is the computation that is involved in it. So I'll try to give an understanding of what goes on behind the scenes when you get genomic data and how do you analyze the data to come up with copy number variants specifically. Many of you might be familiar with point mutations and SNPs and how to detect them. That's largely a string matching problem. But copy number variation detection is a little bit different, so I'll spend more time on the copy number variation detection part of it. Okay. So as a broad outline, we'll briefly cover the different types of variants that we'll analyze in a particular sample. Then we'll focus a lot more on copy number variant detection. And there is one standard method that is used for copy number variant analysis. It's called depth of coverage analysis. And it can be used in different permutations and combinations for different applications. So I'll spend some time on depth of coverage analysis. And then it's not like all of these techniques are exclusive to each other. You can also put together different techniques to solve individual cases. So I'll come to that in the very end. Okay. So the types of variants, all of us know that there's a reference genome against which we compare always, and any difference with respect to that reference genome is what we call a variant. So let us say we have a reference genome from NCBI, we just downloaded it, and you have the entire reference genome. So if you could sequence an individual and you could just sequence their entire genome using long range sequencing, if you could generate the entire sequence, then you just have to compare the reference genome with the specimen genome, S is the sample. So you're comparing the reference genome that you have with the sample genome that you have, and you're trying to understand what are the differences between these two strings. And here you'll try to, there's different ways of saying what is the smallest, you always need to quantify what does it mean to be the smallest number of differences between the two sequences. But let's assume you've done that. And then if you've done that, then you have something that is called an alignment, which is showing you exactly what are the differences between the reference genome and the sample genome. And then you look at those differences and say here there is a T in the reference genome, there is nothing in the sample, so it means that this individual has lost that T. And that is what you would call a deletion. And then here there is an extra C that is there in the sample, which is not present in the reference genome. So that is an insertion that is happening in the sample, which is not there in the reference genome. And then finally here the reference genome has a G, whereas the individual has a C, so you'll call that a substitution. So pretty much all of the small variants until 50 base pairs or so, you can identify it by simple string matching algorithms. Now, how do you generate the string itself, and is it long enough or not, and how do you do the comparison at the right location of the genome? All of those are different other topics, but I'm not going to delve into it, because fundamentally you have a string, you're comparing the strings, you're looking at the differences, and you're cataloging them as deletions, insertions, and substitutions. So these are what we'll call small variants. The next type of variants that you have is called structural variants, where you have the reference genome. I'm showing you two chromosomes of the reference genome. Let's say there's chromosome A and there's chromosome B, and I've just made some arbitrary blocks of the reference genome. So the reference genome, the chromosome A, I'm just arbitrarily putting 3 different blocks in it, A1, A2, and A3. And the second chromosome B has B1, B2, and B3. But in this particular individual, for some reason, the A2 that was there on chromosome 1, in this individual, that A2 has gone to the second chromosome, and it has integrated itself in a flipped manner. So essentially, the entire genomic content is present, but it's not really present in the same order. One part of one chromosome has gone to another part of another chromosome in a different orientation. It could have gone within the same chromosome also, it could have gone to a different chromosome, it could have gone in the same orientation, a different orientation. whatever it is, these are what I call structural variants. And in this particular case, you would call it an inverted interchromosomal translocation. Yes, the name is fairly self-explanatory, right? So this is the second type of large events that can happen. And when these events happen, if there is any gene that is straddling across the boundary, right? If there was some gene that is going across this A1, A2 boundary or the A2, A3 boundary, that is the gene that would get disrupted because of this genomic sequence moving, and those are the genes that might get disrupted and might have an effect. Okay. So these are called structural variants, but I'm not going to focus on these also. The third type of variants that we typically look at are copy number variants. So again, let us say you have the reference genome and you've just arbitrarily broken it up into regions or blocks, and say that the reference genome is a concatenation of R1, R2 to R9, let's assume that's the reference. Then in a normal cell, you would expect 2 copies of each of these because you have 2 copies of each chromosome, one from the mother, one from the father. So you have 2 copies of region 1, then you have 2 copies of region 2, all that is what you have in the normal genome. But in a particular sample, maybe you don't have exactly those same number of copies. For region 2, you have only 1 copy. For region 3, you don't have any copy of it at all. Region 6, you have an extra copy, and region 7, you have 2 extra copies. So the genomic sequences are the same, but you have different number of copies of these sequences, so that is called a copy number variant. So again, these blocks, I just drawn them for illustrative purposes. It doesn't mean they're equal sized or anything. It is just a representation here. And the way you would represent this in the final algorithm output that comes out of copy number variant analysis is to say that I have a region 2, and you'll have to specify the coordinates of that region. It might be chromosome 15 from 50 megabase pairs to 60 megabase pairs. Something like that would be the definition of the region. And so region 2, I have only 1 copy in the sample, so it's a heterozygous loss. Region 3, there are 0 copies, it's a homozygous loss, which means it's a loss on both copies of the chromosome. Region 6, I have 1 extra copy, the copy number value is 3, so it's a 1 copy gain. And region 7, I have 2 extra copies, that's a 2 copy gain. So the output of a copy number variant algorithm on a particular sample would fundamentally be this table that is shown at the bottom, right? So now let's look into copy number variants in more detail and say what is the method that we use typically to come up with this particular table. So all of you must be familiar with this picture. You have 2 copies of each chromosome and you have a banding pattern. It's called a karyogram. And as a notation at the bottom, they'll indicate by what is the total number of chromosomes. There are 46 chromosomes, 44, 22 times 2, 44, plus either an XY or an XX. So the standard way of representing, summarizing this entire picture is that I have 46 chromosomes and XX for female or XY for a male. Right? This part is clear. Now, if the copy number changes are extremely large, and the largest copy number change that can happen is if the entire genome itself, you have a 3rd copy of it. So that's called a triploidy. So instead of having 2 copies of each chromosome, you have 3 copies of each chromosome. So now you have 69 chromosomes in total. And in this particular case, it is an XXY. So that would be the notation, 69XXY. So if you had this larger change, you could detect this change through microscopy. You could take the cells, you could culture the cells, you could freeze it at a particular point in cell division, you could look at it under a microscope and say, aha, instead of 2 copies, there are 3 copies. I'm seeing that for every possible chromosome, and I see 2 X's and 1 Y, and you say it's a whole genome triploidy event. So this is the largest possible copy number alteration that you can see. But in reality, most of the chromosomal copy number changes are far smaller than this. So you can't really see them under a microscope, and that is where sequencing comes in. So what is the basic sequencing based workflow? In our lab, we'll get an amniotic fluid sample or a blood sample or some other sample. From the sample, we extract the DNA. Then the DNA has to be sheared into smaller molecules before it can go into the sequencer. We shear it typically into molecules of size between 200 to 700. And then we load that onto a sequencer. The sequencer will generate fixed size reads. Sequencer, I'm not talking about the long read sequencing, I'm talking about short read sequencing. So we'll get around 150 base pairs of each fragment. So those are called the reads. So each one of those lines that you see in the right hand corner that is a 150 base pair ACTG sequence representing some fragment which came from somewhere in the genome. We have no idea where it came from. So that set of reads represents the entire set of genomic sequences that were derived from somewhere on those 23 chromosomes. That part is clear, right? So now we take all of that strings that are out there, and now we have to match that against the genome to say where did each one of those reads come from. And we do that, and then that will be called alignment. So each one of these lines out here is one of those reads that came out of the sequencer. And we map it to the genome, and you see it. And in this particular case, it was a 40x whole genome sequencing, which means that if you were to look across the genome, at any place that you look, you will see roughly a depth of 40. And this is called the coverage track. It is showing you how many reads are there at this particular location in the genome. So we started with an ordinary sample where you have 2 copies of each chromosome. We shredded it into small pieces. We sequenced it. So we expect to get the same amount of data from all at any point in the genome. And we mapped it. And we can see that we have the reads and the coverage. And we can see that the coverage is fairly uniform around 40 all across the genome. Okay? This is what it will look like in a normal sample. But if there had been a region out here, let us say, which had a copy number gain, then the molecules, instead of coming from 2 copies, would be coming from 3 copies. And what you'd see is that this coverage would shift up and you'd have more coverage there. Whereas if there was a copy number loss, 1 copy had been lost, then you'd see only half the coverage at that particular location. So fundamentally, you're looking at the coverage, you're looking into variations in the coverage pattern. From the variations in the coverage pattern, you're trying to infer, is there a copy number gain or is there a copy number loss? So that is why in all of these methods are called depth of coverage based analysis. At the same time, you could ask, even though you're supposed to have around a 38 coverage out here, at this location, the coverage seems a little bit low. At this location, the coverage seems a little bit high. Is there just a stochastic noise, or is there an event happening out there? Is it possible that there's a small deletion happening out there? Is it possible that there's a small amplification happening out here? Things like that. Now, the way to answer that question is to say, what is the underlying biology that we're trying to understand? So, for instance, there is a notion of mosaicism, which means not all cells may be the same. So, in a particular individual, there might be 10% of the cells which are slightly different from the remaining 90% of the cells. If there was a copy number gain on only those 10% of the cells, then the signal out here would not jump by 50% as it would if it were a third copy. If you had a whole region having an amplification, instead of having 2 copies, you would have 3 copies, it will be a 50% increase in the signal value. But if it's a mosaic event, only 10% of the cells have that event, then the signal value would jump only by the 10%. So you can't really detect mosaic events of very fine granularity from here. But if you had events of 50% increase in the number of regions, 50% decrease in the regions, so it's broad signals is what you can find out. So hopefully everybody now understands what depth of coverage analysis is. So now let's look into the actual algorithms that use this. So again, all the algorithms will start with this coverage profile. Now you can't really analyze every single base and say is there a copy number gain or loss, it's just too much. So you'll have to make this data more discrete. So what you'll do is you'll just put some arbitrary bins along the entire genome. And here, for instance, I've divided this stretch of the genome into 11 bins or regions. Okay? So each one of them will have a chromosome start and end, things like that. And the first thing I can do is I can look at the reads in that particular region and say how many reads are there overlapping this region. So the first region, there are 2,000 reads out there. The second one has 1,800 reads. I just convert that coverage plot where for every single base I have a coverage value. I convert it into for each region, how many reads do I have? Now this number of reads that I have out here is a function of how many reads I generated in my experiment. Maybe I generated 15 million reads and therefore I'm getting this. If I generated 40 million reads, I would get 2.5 times this. So next thing we do is normalize this using some factor and say that if you were really expecting 1, let's assume if everything is 1, it means there's no change in copy number at all. With respect to that, how much deviation am I seeing? Now the first region it's 1, the next one is 0.9 and so on. So now you have a normalized count for each region, and each one of these deviations from 1 could indicate a copy number gain or a copy number loss in a small fraction of the cells. Oops. So if you have this 0.9 out here, it could be because of a genuine copy number loss in 10% of the cells, or it could just be noise. Now to distinguish the two of them, what we do is we do the sequencing for 30, 40, whatever it is, number of normal individuals. There's nothing like a normal individual, healthy individual, let's call it. We generate this data for a set of control samples, and then from them we calculate the mean and standard deviation of this normalized count value for each one of those regions. And let us say you have the mean and the standard deviation. And then we say that is the signal significantly different from what we expect based on the profile that we have constructed. So this is what we would call a profile. So here, for instance, the first bin in the profile that you calculated from across, let's say 100 individuals, the value mean was 1.1 and the standard deviation is 0.1. So in our sample, we are seeing 1.0. That's not really a big difference. So you would not really call it out as statistically significant. Whereas in the second one out here, the signal is, the profile says 1, the sample has 0.9. So if you take, x minus x bar by sigma to get the z score, you'll get 0.1 divided by 0.01. So the z score would be 10. So it's fair, because the variance is very, very low in this particular bin, it's only 0.01, this is still a significant value. And that is how you call out which regions are potentially losses, which ones are the potentially gains, and you call out all of these events. Okay? I'm not saying we'll do this exactly, but hopefully you got a flavor of how the depth of coverage analysis is used to call out copy number events. Now you have two copy number events, both of them are losses which are right next to each other in adjacent bins. You'll probably merge them and say that it's a larger copy number event, right? So you'll try to combine events of the same type. Now there'll be all kinds of complexities, saying you might have gotten 4 copy number losses, then there was a normal, then there were another 4 copy number losses. Is it really 2 different events that are happening separated by a small region? Is it just a statistic? Should you merge all of them into a larger region? Those are all the next level of details that we need to sort out. Okay. So this is a basic algorithm, and this basic algorithm is used in a million combinations. Every paper will have some flavor of this with a different set of parameters for it. The first thing obviously is what regions to use. Should I be using 1 MB size regions? Should I use 2 MB size regions? What regions should I use? Should the length of the region be fixed, 1 MB all the time? Or depending on the sample, should I change my region size? Because if I'm sequencing to a very high depth, then I can afford to have small regions. But if I'm doing only 1x sequencing, doing a very low pass sequencing, then my regions will have to be larger. So how do I define that bin depth, bin size? The second thing is, should all regions be of the same size? Or should I say that this region for some reason gets a lot more reads? I'll keep a small bin here. This other region gets very low number of reads for some reason. I'll create a large bin here. Those are variable things. Then we also need to take into account that there's a lot of homology in the genome, which means there are lots of duplicated sequences, and then you can't really trust the value there, so maybe you'll exclude those bins. Those come in. And then after you have mapped the reads to the bin, should I treat all reads the same? Some of the reads might have mapped with very poor confidence. Should I weigh them separately? All of those come in. And then should I just start with the normalized counts, or should I do some additional correction? And I'll get into that in the next slide. And finally, after I've generated my normalized counts, what is the profile that I'm comparing it against? I mentioned that there's a mu and a sigma and a profile. Should it be a gender specific profile? And that's what we do. We'll have a male profile and a female profile, you'll have it. But you could have any number of profiles. The blood samples behave in a certain way. Cheek swab samples, for whatever reason in the laboratory, they introduce a different type of signal. They behave slightly differently. Amniotic fluid samples behave slightly differently. Every batch of samples that we sequence on the sequencer, for whatever reason, they'll have a slightly different normalized count profile. How many different things should we take into account to compare one sample against a profile? What is this profile? That becomes the next thing. So what we do in clinical practice is that we look at a particular sample, we look at its characteristics, we consult the database of all samples that we've done in the past, we identify similar samples, and we use the similar samples to create a profile to compare an individual sample against. So it's computationally far more challenging, but it helps to reduce the false positives a little bit. Okay. And then how do you combine nearby bins? If you have 2 events separated by 1 MB, do you merge them? If they're separated by 5 MB, do you merge them? all of those nitty-gritty details. So I won't go into many of those complexities, but one of the most important factors is GC correction. So I'll just talk about this one factor. So as you're aware, when we create all of those bins in the genome, every bin has a certain GC composition because it's referring to a particular portion of the genome. You can say what is the percentage of G and C nucleotides in that particular portion of the reference genome. That is called the GC percentage of the region. And different regions, so here we are looking at, we sequence the exome, so you have around 220,000 regions. So, and every region will have a GC percentage. So here I'm just plotting for every GC percentage, what is the coverage profile of the regions of the GC percent. So here for 52, maybe there were 1,000 or 1,500 regions which had a GC percentage of 52%. And I'm saying what is the average coverage that I see in those regions. And this is a well-behaved sample because even if the GC percentage is varying widely, the coverage is fairly uniform. But you will also have cases like this where a sample, for some reason, the high GC regions got a lot of data, the low GC regions got a less amount of data. So this is not copy number change. This is just an artifact of the laboratory process because of which more data got generated from the GC rich regions, even though the copy number was same all through. So you'll have to do some correction of this GC. It has to be taken as a factor into normalizing the counts before you do the copy number analysis. Okay? If you don't do that, then what will happen is, if for instance I had used this as the profile sample and this as the clinical sample I'm comparing, then all the high GC regions would have been called out as copy number gains. All the low GC regions would have got called out as copy number losses, even though in reality nothing like that was happening at all. So this use lowest regression and other methods to correct for GC correction before you do the depth of coverage analysis. Okay. Yeah. So now that you got the basic techniques in place, now let's look at some examples of how we have used copy number analysis tools for different applications. One of the first applications we've been using this for 10 years, but last year we were doing whole genome sequencing at low pass. So instead of generating that 40x coverage data, generate only 2x coverage data across the entire genome in an effort to identify very large scale events. And the extreme case is if I were to use only one bin for chromosomes. So in other words, you take all your 20 million data points and just come up with one number saying how many reads are there for each chromosome. If you do that, now I won't go into this, and at the time we're developing this test, I was teaching 10th grade chemistry to my daughter, and it struck me that there's a perfect analogy between analytical chemistry where you're looking at the empirical formula of compounds, you have the percentage of carbon, percentage of oxygen, and you're trying to find out what is the molecular formula. And exactly the same thing is happening if you have one number for each chromosome. You're trying to come up with chromosome 1, 2 copies, chromosome 2, 3 copies. It's exactly the same molecular formula. References in the analytical chemistry book. So if you do that and we plot it for 100 samples that we have done, amniotic fluid samples, without doing any GC correction, without doing anything, you get a beautiful signal, right? So these are all Down syndrome cases. You have chromosome 21, 3 copies. There's a chromosome 15 here. And you remember the karyogram that I showed before, that 69XXY? It shows up beautifully. You have 3 copies of all of the chromosomes and an XXY. Okay? So sometimes if you have very large scale events you're looking for, the simplest methods will also work beautifully. But then you have, if you want to find finer grain events, these are 250 kilobase per bins, 2x coverage. Then using the same methods, you can call out smaller losses and gains. And these are for cancer samples, fundamentally using the same method. Because of times of limitation, I'll kind of skip through this, but you get a flavor of what is going on, right? So basically, each application requires its own type of analysis. So for prenatal diagnosis, amniotic fluid, you're looking for whole genome sequencing course. You're looking for fairly large events. In rare disorders, where somebody is already, a child has a rare disorder, you're trying to find an event. The event is likely to be far smaller because very large scale events lead to death very early on. So usually for earlier in the reproductive health part, you're looking for large events. Later, you're looking for relatively smaller events. I will skip this section in the interest of time. Yeah, some other day I'll get into this, but I'll skip that. So we talked about point mutations and we talked about copy numbers, and sometimes you need to look into both of them to solve a particular clinical case. And this is a case for which we released the report only last night, and it was a particularly interesting case, so I thought I'll go into it. It's a girl child, 16 months old, right from when, from maybe last year sometime, they noticed epilepsy, there was some problem with eye movement, there is dystonia, things like that are going on, and they have already got a sequenced exome sequencing done. They found a mutation, but it didn't fully explain everything. They found a mutation in one copy of a gene, but the geneticists felt that, even with one functional copy, the child should have been okay. So only one pathogenic mutation in one copy does not explain it. They did copy number sequencing, they didn't find anything, and then finally we sequenced the child, the mother, and the father, all three of them together, trying to identify the segregation pattern. So I'll take you through this a little bit carefully. It's the last slide, but it'll take some time. So you're familiar with the coverage pattern, right? Saying how many reads did we get in each part of the genome. And this was not a whole genome sequencing, so these are only the exons. That is why you see some discontinuities. So I'll be showing multiple panels. The child is on the top, then the mother, then the father. If you were to look only at the coverage pattern, it's kind of hard to figure out exactly what is going on in the coverage pattern side. Then we did the variant calling. Hopefully you're able to see that these are blue and these are red. The blue are heterozygous variants. The reds are homozygous variants. Nothing to do with pathogenicity, just heterozygous, homozygous. And we found one heterozygous deleterious variant in the child and the father. It's not there in the mother, which means one copy of the gene is most probably knocked out in the father, but the father is asymptomatic and it does not, which means this alone cannot explain what is happening in the child. The child inherited one bad copy from the father, but that alone does not explain everything. And then we did the copy number analysis, and this shows the copy number track. This line is the copy number 2 track, and here indicates it's a deletion. Okay? This is the copy number 1. You can see that this portion of the gene, 10 exons of the gene, have been deleted in the mother and in the child. Okay? So this is what you call a compound heterozygous case. The child, the father had one deleterious mutation, but the other copy of the gene is functional in the father, so it's okay. The mother has one copy number loss event, so the mother also has one non-functional copy of the gene, but the mother is also okay. The child unfortunately has inherited the point mutation from the father, so that knocked out one copy of the gene. Child inherited the copy number loss event from the mother, that knocked out the other copy of the gene. Okay? So sometimes we need to put all of these things together to be able to explain a clinical case. I think the report would have gone out this morning. I think for almost a year they've been trying to look for a solution about what is happening. And this required a huge team effort. The geneticist understands what genes are important. At the same time, when one hit doesn't explain it, they're asking us to look for a second hit. Multiple labs have looked at it, and then you put it all together, and luckily we found something. Obviously, we don't know how to solve many of these rare disorders, but at least it provides an answer to parents who are looking for understand what is going on, right? So CNV analysis, conceptually simple, just depth of coverage, but all of those different batch effects and binning and parameters, that's what makes it complex. And detection of these events is just a start. We find a lot of these events, but explaining, do the events fully explain what the doctor is seeing in the patient or not, that is also very, very important from a clinical perspective. So in our company, it requires coordination between the lab people to say, was there something odd when they process the sample? Should we be worried about the aberrations or not? The bioinformatics team, the variant science team, the genetic counselors, interaction with the clinicians, it really is a team effort. And so the slides are based on the clinical data I had. Thank you all for your attention.
Thank you, Dr. Vamsi. Questions? Lots of questions. Start with Ruth.
Wonderfully detailed talk. So couple of questions. When you say as you said, it cannot be normal, healthy individuals. So how do you profile the healthy individuals? And secondly, when you have blood samples from males and females, I'm not talking about the other types, but DNA isolated from whole blood, do you really see a difference in the profiles between males and females?
Yeah. So I'll take the second question first. We don't really see a difference, but because we also call out X-linked variants and all of that,
Okay,
just
for
for
that.
just for
Okay.
that part of it, we keep
Okay,
it separate.
okay, yeah, that
Otherwise,
makes
no,
sense.
it doesn't make a difference.
Okay, yeah, that makes sense.
And when you talk about normal, so all of the data that we process, it goes into a central database. So every variant that we find, we compare it now against 10,000 exomes that we've already processed in the laboratory. And we are looking at rare Mendelian events, right?
Okay.
So we don't expect it to be common in our database of 10,000, and that forms the normal. So for any individual gene, there's a lot of normal, though every individual might have a lot of abnormal
Right,
things.
right, yeah. Okay, okay, got it.
Yeah.
Thank you.
Hello, sir. So, is it possible to analyze coverage, in-depth coverage analysis for gene-specific?
Yeah, so there are two types of sequencing. There is whole genome sequencing and there's targeted sequencing. So the clinical data I showed you in the last slide is actually targeted sequencing, where we sequence only the exons of the 20,000 coding genes, but we can make smaller panels also. So we do germline cancer where you do only 94 genes. There is some other panel where you do 44 genes. So if there is some particular study where your focus is only on a few genes, then you can always create a panel for only those genes and do depth of coverage analysis for those.
Okay, so it can also be applied on plant as well, right?
You could do it on plants also. Now here, most of the analysis implicitly had that assumption there are 2 copies. In plants, you'll have sometimes 6 copies of a chromosome, I think in the grasses and
Yes,
all it's
sir.
there.
Yes, sir.
And you'll have to see, can I detect a change from 6 copies to 7 copies? The signal will be a little bit more muted. You need to, and to control that variance, you'll have to generate at a higher coverage.
Okay.
That stat
So
you'll have to work
based
out.
on the ploidy of the plant, I can do it.
Yeah.
Okay, thank you, sir. Very informative.
So I have a question, and out of curiosity. So, what kind of symptoms would a person with, triploidy present?
Oh, triploidy is lethal. So you get it. So we get these requests from patients who mothers who are going for pregnancy screenings. So at the 16th week or 12th week and all, they have gone for some ultrasound, regular ultrasound checkup. And if at that time they feel that the fetus is not developing properly, they see some gross abnormalities, then they would take an amniotic fluid sample and send it to us to decide whether to continue the pregnancy or not. Triploidy is not going to be
I see.
viable.
Okay, another question. So, I guess in this CNV calculation, you're masking the repeat regions, right? Are you?
Now it all depends on that will get into. You need to be consistent between your profile and your clinical sample.
Okay.
So if you didn't mask it in the profile and you're anyway comparing it against the profile of 10 or 20 people that you already sequenced, then it may not matter.
But this consideration can be ignored if I'm having long reads?
If you're having long reads, most probably you could ignore some of these things. But I don't know what all, just the way that we are familiar with GC bias and a lot of other things that happen in short read sequencing, I'm not familiar enough with long read sequencing to know what biases might be there in that.
Thank you.
So can I take the call? Yeah, very nice talk. I know you simplified a lot of things, but I have a question here. I had an earlier paper, and many others also have talked about it, the so-called dark matter, in the sense of intergenic regions, right? So I can talk particularly about mine because I understand that better. We were looking at classification, very subtle classification of cancer, subtypes, and we found signals using some AI methods. We didn't know what to do, how to distinguish, we didn't know how to do, but we tried AI methods to distinguish these subtypes of a specific blood cancer. And it turned out that most of the signals were in the dark matter, in the intergenic region.
Yeah.
And there are similar results coming from others. So are you looking at these things? Have you had cases where you have to look at this dark matter, or?
Yeah, that's a great question. So every diagnostic test, we need to have SOPs for our reporting processes, right? So usually we follow we always follow the ACMG guidelines for recommending a variant. And so there are probably 20 pieces of evidence and individual scores that we need to give to each one of those evidence. And these guidelines came up in 2020, because otherwise there's a lot of subjectivity in what do you consider relevant variant, what do you consider not. So they're based on segregation, they're based on group studies, they're based on animal models, all kinds of every type of evidence that you can give towards why you think the variant is implicated in this has a certain amount of score that you can give it, and then you add all of that up, and then it falls either under a variant of uncertain significance or likely pathogenic or pathogenic. So many of the clinicians that we interact with, they want something that they can give an answer to a patient in a fairly short amount of time. So more interested in likely pathogenic and pathogenic variants. And even amongst the variants of uncertain significance, our internal scoring, we have 5 different segregations in them. So the topmost tier of those variants of uncertain significance, we convey to the doctors and say, do you want it included in the report or not? So many of these intergenic things, because so little work might have been done on them, there may not be enough animal thing, it would not make it to any of those categories, and we would not be able to report on it in a diagnostic context. In a research context, we have looked into some of these. And this month we've had 3 cases where you could not find any answers in the exome, and people have asked us to do whole genome sequencing and look at it. It's turning out to be quite challenging because you'll have so many variants. We'll start getting familiar with it, but in a diagnostic context, it's very hard for it to reach that level of evidence.
Yeah. Thanks.
Hi, very interesting talk. I guess since this is a computational kind of gathering, and you showed various ideas that have already been explored, if you had to pick something, what's the biggest challenge that you still face in copy number variant calling, and what could we still work on which would make a difference to what you're doing right now?
So copy number calling is all about the size of the event and the accuracy. So large scale events, it's fairly easy to call out, but there are, we believe there's a lot of rare disorders where it could be just a single exon that has been deleted. And those are the most challenging things to call out, because a single region out of 220,000 regions, to call that out, I would say is still amongst the most challenging. There's a paper that came out last year where they said great progress made in copy number calling, and I went excited and looked at it. It's around 69% sensitivity and around 55% specificity at calling out these really fine events. In this particular case, we were able to verify it because it was a 10 exon stretch, but where we really struggle is when we see a signal in 1 exon or 2 exons, is that noise or not, because you need a huge amount of data for us to control the variance at that level of detail. We have even reported on case where there was a half exon deletion, where half of it was there. That's because the doctor had an inkling that it might be that gene, and we went and looked at it, but otherwise we would have just missed it through routine processing, right? So if I were to pick one challenge, I would say that at exon level, copy number detection reliably, in the context of all of these GC biases and all of those, you cannot ignore the lab processes at all. You can't say that, somebody has given me the data, I will analyze it. You need to be aware of all of the lab metrics which could have caused batch effects. Taking those into account and calling out single exon events, I would say is the biggest problem we have right now.
So it seems like part of the problem is that you only do the measurement once, and then you're doing the data post-processing. And if one could do it, put a feedback between those 2 steps, you could probably discern finer grained variations.
That's a good point. I mean, for clinical reporting, you need to freeze on your SOP. You need to describe exactly which profile you're going to use, and anytime you change anything, you need to have the documentation trail about why you changed it and so on. So you could think about the profile itself, updating it once in a while, so that, as your laboratory kits and all are changing, maybe the profile is changing slightly, keep updating that. You could talk about sequencing the same sample a second time. Now the same sample, there are multiple stages at which you could pick up that sample for sequencing. You could go all the way back to the blood and extract it, the DNA fresh. That way you're eliminating even the extraction batch effects, or you extracted the DNA, you made a library out of it. Which point do you start from? All of those are there, and India is a highly cost sensitive market. These tests, there's no profit in them.
Hi, it was very well detailed talk. So, if I'm not mistaken, you are calling CNVs based on coverage depth, difference in coverage depth. So, is there any other factor than the CNV present which can affect the coverage depth? And if yes, how are you dealing with that?
So there are two parts to that question. One is we are calling copy numbers only using coverage depth. Those were the missing 4 slides that I skipped over. You can also call out copy numbers using change in the allele frequencies. Right?
Yes.
I mean, a SNP would have a 50% allele frequency. There's 2 copies, but if there are 3 copies, it will become 33% or 66%. We use that in some of our cancer analysis. We don't really need to use it in germline cases because it's fairly clear. That is one part. The second one is saying what all can affect coverage. A whole bunch of factors can affect coverage. The number of PCR cycles that you use to process the sample. If there's a temperature difference there, then the GC sequences amplify more or the AT amplifies more, and that induces changes. There's a slew of factors there.
Thank you.
Right, Vamsi, I'm going to ask one last question. I think we need to stop. One last question, which is, you mentioned about large deletions or large insertions using depth of coverage. That's easy, but when you have, medium-sized insertions or deletions, it becomes hard. In those cases, do you think that microarray data will be more useful than depth of coverage data? Our experience has been that, for medium-sized microarray data, it's easier to call, but I don't know what your...
Yeah, so we worked on this only in the last 6 months as we released CNC copy number sequencing through whole genome sequencing products. And you're right, it depends on what is the depth of coverage to it, through which we do this. Some of the requirements are only for 500 kb and larger, and in those cases with 1x coverage we're able to call it out. And recently we're calling it out at 25 kb and larger, I think, with 4x coverage. So I think if you can modulate the amount of sequencing coverage that you do, whether it is 1x or 2x or 4x or 5x, if you can calibrate that, then different ranges of size of events can be called out. We did a comparison with what had been done with microarray, and so far we have excellent agreement with those.
I don't want to be rude, one last question.
So you mentioned that in the genome we'll select some regions in arbitrary ways. So there can be many ways to select the regions. So what is the best way you find to select the regions that suits?
Yeah, when you do whole genome sequencing, it's always, if you don't have any other criteria, the simplest is the best. We just take 1 MB and just tile the whole genome with consecutive 1 MB or 5 MB regions.
Thank you.
Yeah, thank you all.
Thank you very much.